This video is the third in a series about nucleotides for OCR A-level biology. Living organisms need to produce a really wide variety of proteins, everything from globular proteins like enzymes and hormones through to structural proteins like muscle fibres. And for each one of these proteins, the organism needs a recipe, and this is written in the genetic code, which is DNA. DNA contains 4 nitrogenous bases, which form the basis for this code, and these can be arranged in any order. In order to translate from the DNA code to the sequence of amino acids that make up a protein, we use a system that consists of triplet, non-overlapping, degenerate, and universal code. To help us to explain what this means, let's look at a length of DNA. This is the sense strand of the DNA. In other words, it's the piece of the DNA that actually gives us the sequence for a gene and that tells us how to make a particular protein. It's read from the 5' end to the 3' end. And starting from that 5' end, we can divide the DNA into codons. These are sequences of 3 bases next to each other. The first of these codons for a gene is always going to be methionine, and this is the amino acid that functions as a start codon. This part of the DNA sequence is non-redundant or non-degenerate, and what this means is that methionine is always coded for by ATG. There's no alternative sequence. As we carry on reading our DNA sequence, we can see that this particular sequence would code for serine, glutamine, proline, arginine, threonine, serine, glutamine, and then a stop codon. That stop codon is redundant. There are 3 different codons that all code for stop. We've described what triplet means, that idea that 3 bases are needed to code for 1 amino acid. But what about non-overlapping, degenerate, and universal? Well, non-overlapping means that when we're reading this particular strand of DNA, we're going to read ATG as being 1 amino acid, and then AGC as being a second amino acid. We wouldn't read ATG and then TGA, because those 2 codons would then be overlapping. When we describe the genetic code as degenerate or redundant, what this means is that there are multiple different DNA codes that code for the same amino acid. So for instance, to code for glycine, there are 4 different codons that I could use. And then a universal code means that in every organism, the same DNA sequence is going to code for the same amino acids. The process of protein synthesis can be broken down into 2 stages, transcription and translation. Even if you haven't met those words in a biological context before, you've probably heard about them in everyday life. Transcription means writing down something that someone else has told you, and translation means moving it into a different language. And this is just what we find with protein synthesis. Transcription is copying the DNA code, and then translation is putting it into a different format, namely protein. The reason that we need this transcription stage is that DNA is too large to leave the nucleus. So therefore, each time that a gene needs to be expressed, a smaller section of the DNA is transcribed to make messenger RNA instead. DNA consists of a sense strand, which gives us the code for the protein, and then an antisense strand, which is the complementary sequence which runs alongside it. But when it comes to making the protein, it's actually the antisense strand which is used as the template, because this means that when the mRNA forms, it will have the same sequence as the sense strand. In order for transcription to take place, the mRNA nucleotides need to be able to get alongside the gene that needs to be expressed. So the first step in transcription is that the DNA unzips from the start codon. DNA helicase achieves this by breaking the hydrogen bonds between the complementary nitrogenous bases. Next, free RNA nucleotides line up alongside the complementary bases on the antisense strand. Phosphodiester bonds are then formed by RNA polymerase, which moves along from a 5' to 3' direction. Once the mRNA is fully formed, it detaches from the antisense strand of the DNA and leaves through a nuclear pore. The DNA double helix then reforms. Pause the video and see whether you can complete this description of protein synthesis. Firstly, the DNA unzips, starting from the start codon. The enzyme that breaks the hydrogen bonds between the antiparallel strands is called DNA helicase, and this allows the DNA to unzip. Then free RNA nucleotides line up alongside the complementary bases on the antisense strand. Phosphodiester bonds are then formed by RNA polymerase. The mRNA then detaches and leaves through a nuclear pore. And then the DNA double helix reforms. Now is a good time to pause and remind yourself of a few key differences between the structure of DNA and messenger RNA. DNA is always double-stranded, where messenger RNA has a single strand. Obviously, DNA contains deoxyribose, where mRNA contains ribose. When it comes to the code, both nucleic acids contain guanine, cytosine, and adenine, but thymine in DNA is replaced by uracil in mRNA. And DNA is obviously much longer, being millions of base pairs long, compared to mRNA, which will be from a few hundred to a few thousand base pairs, depending on the length of the gene. Translation is carried out by the ribosomes, which use the mRNA sequence to assemble the sequence of amino acids that make up a primary peptide. Ribosomes contain two subunits, one large and one small. These contain both protein and ribosomal RNA, known as rRNA. The mRNA binds to the small subunit of the ribosome. As well as mRNA and rRNA, there is a third type of RNA called transfer RNA. Transfer RNA, or tRNA, binds to the mRNA via the complementary codon. So the mRNA, remember, basically matches the sense strand of the DNA, with the exception of the fact that every thymine has been replaced with a uracil. The transfer RNA has a triplet codon at the bottom, which is referred to as the anticodon, because this is complementary to the codon that we see in the mRNA. As each tRNA is placed in order, the amino acids held by each tRNA are placed in the correct sequence for the protein. As these transfer RNAs are added, a condensation reaction is used to produce a peptide bond that joins them together. Ribosomes move along the DNA, and as they release the first tRNA, they recruit another tRNA, and the ribosome will continue to move until it reaches a stop codon. Now pause the video and see whether you can complete this description of translation. Transfer RNA binds to the messenger RNA via the complementary codon. As each tRNA is placed in order, the amino acid held by each tRNA are placed in the correct sequence for the protein. Peptide bonds then join the amino acids together. The ribosome moves along the messenger RNA. It releases the first tRNA and recruits another tRNA. The ribosome continues to move until it reaches a stop codon. Translation by the ribosomes produces the primary structure of the protein. This is the sequence of amino acids that have been joined together and is determined by the sequence of the DNA. However, in order for us to get a functional protein, further folding and modification is required. You should also be aware that when it comes to translation, it's very unlikely that an organism needs a single copy of a protein, and therefore to speed things up, simultaneous translation is taking place. Multiple different ribosomes can be building a protein based on the same mRNA sequence at the same time. Thanks very much for watching, and I hope you're now feeling slightly more confident about describing transcription and translation for A-level biology. If you did find this video useful, then don't forget to like, subscribe, comment and share the channel, and I'll be back soon with some more A-level biology revision videos.